Okay, so here we got a picture of Darwin's Tree of Life. He speculated since species aren't fixed, we could probably reconstruct a family tree for all organisms. So that's phylogenetics. And this was a new effort in the taxonomy or classification of organisms. People have been doing this for centuries. The ancient Greeks grouped all the organisms and they figured there were maybe 500 species. Linnaeus in the during the Industrial Revolution knew that there's thousands and thousands of species, and so he developed the system of binomial nomenclature, where each species is given a Latin name, two-part Latin name, genus and species, and then further divided up into related groups based on homologous structures that we think that they shared based on common descent, so how similar they are. Later, much later in the last century, they realized, well, this isn't enough. um, Linnaeus kind of divided things into five kingdoms: animals, plants, fungi, bacteria, and the single celled protozoans or protista. And they really realized, well, the bacteria, what he called Monera, was really two different types of organisms, and they were added as domains. And so um, four of the five kingdoms became eukaryotes, and then two domains of single celled bacteria like organisms, bacteria and archaea, were added later. Okay, so we've talked about phylogenies, and that this is an important idea for comparative biology. Looking at homologous structures derived from the same ancestral source, you can identify organisms that are related. Okay, it's important to kind of weed out here structures that are analogous or homoplastic. They look the same, but they're not the same because of descent. So, like bird wings and dragonfly wings, um, they both are used to fly, but neither are related to the other. So, in the phylogenetic analysis can help determine whether a structure is analogous or homologous. So here we have a little bit of um, a, a cladogram there that's showing, okay, um, there's features there that are shared by organisms, and here we can see the family tree of this. Okay, and so that's a monophyletic group there, and those shows the features that help us decide. Okay, and a larger one kind of classifies these as archosaurs. So our binomial nomenclature system is I don't want to say it's breaking down, but it's changing rapidly. It's being very much modified by Darwin. And so all organisms are being put on these phylogenetic trees using the techniques that we talked about, shared characteristic and homologies. But here we're getting into more and more details because we're looking at cell types, particularly the chemistry of the cells and DNA sequence. Okay, and so we've got this uh, tree here, the rooted phylogenetic tree that shows eukaryotes in red, archaea in green, and bacteria in blue. And so that the oldest organism here is probably a bacterium. Okay, now there's a fly in the ointment because it's so far it sounds like this all works really well. Kind of. There are things that screw it up. Okay, not all genes have been inherited in the same way. Certain genes have been transferred horizontally between different species, sometimes between different domains. And this turns out to be a really important process. Horizontal gene transfer are the same mechanisms that we use to transfer genes in genetic engineering: the use of plasmids, transformation, transduction, using viruses. These, these techniques can occur between unrelated organisms, not just parents and their offspring, and they they don't transfer huge numbers of genes, and they happen kind of rarely. But over a long period of time, these could be very, very important uh, mechanisms, and they can transfer and give organisms new traits. And we think this is a major event in evolution. In fact, we think that their major groups appear to have arisen via these horizontal gene transfer mechanisms. In fact, us eukaryotes appear to be mosaic organisms, partly arisen by uh, inheritance from a line of ancestors, but also from gene transfer. Okay, endosymbiosis may have given us um, the mitochondria. We think perhaps gene fusion has uh, is the explanation for why we have a nucleus. We can also see that in other organisms, like in gram-negative bacteria. They have two cell membranes, and one is a cell membrane that's clearly from a gram-positive bacteria, and the other looks like it's probably from a fused gram-negative bacteria, or an endosymbiotic relationship between gram-negative and gram-positive. So the history of life, it's going to be even harder to tangle out than Darwin thought, because we have this, and so there are some other ideas that may we don't know, that we may think the three domains came from some of these horizontal gene transfers. In some cases, we think eukaryotes arose by an archaea that swallowed a bacteria that became the mitochondria, or a bacteria that swallowed another bacteria and then swallowed an archaea. Or some guys have even proposed that the eukaryotes were the first organism, and that they they kind of became simplified to give off bacteria. And these are kind of strange, mind blowing ideas, but we do have evidence that we can evaluate here. And not all this evidence is homology structure evidence, much of it is fossil evidence. And so the fossil evidence certainly suggests that bacteria are older than eukaryotes. Okay, we have bacterial fossils, stromatolites, that are 3 billion years old. There's no fossil eukaryote that's older than a billion, and very few that approach even that age. We can also look at bacteria and think they're far more diverse metabolically. They can do chemical reactions that eukaryotes cannot do. The um, oxidation and reduction of sulfur and sulfates, of nitrites, nitrates, using iron as a source of energy, um, uh, methanogens, all these kind of metabolic diversity couldn't have evolved without lots of time, and so they've got to be older than eukaryotes. And and so you have to use parsimony and kind of these other pieces of evidence. But no matter what you think, the history of life is far different than what Darwin originally thought. We know that that life goes at different paces for different organisms. That Darwin's simple tree is at best a networked or branch tree, and the ring of life idea is that maybe horizontal gene transfer was really important, in that the primitive pool of prokaryotes shared genes pretty regularly. Well, anyway, so what good is this? So this turns out to be important, kind of telling us where to look for life when we go to other planets, when we're looking around in different places on Earth. It also tells us about important things about living organisms that may be important to us. Um, and a good example is with disease organisms, like a virus like HIV. HIV is the virus that causes AIDS, and like all viruses, it reproduces once it gets inside a host cell. And so it has some receptors on its surface that grab onto the host cell. When it's in the host cell, it uncodes its genome, which in this case is RNA. It then copies that RNA into DNA and inserts it into the host chromosome. So the enzyme that does this is a viral enzyme, reverse transcriptase. And then once it's in the host chromosome, it kind of tricks the host cell into making new viruses. And so this thing doesn't have a lot of things that we can compare it with other cells, but you know, we can ask similar questions about it and say, where did it come from? How did it get into people? And can that help us figure out how to do something about it and where it might come from? And and so this question, where did HIV come from, is important. We first saw HIV in the 1980s. Blood samples have since showed that it was much earlier, at least in the 50s, but maybe in the 30s or even before. And that the geography of this was that the early infections were associated with often with Africa and sailors. And then when you looked at sequence of the gene that is most central to the virus, you find that that's similar to the gene that's found in various types of small monkeys, particularly manga bees. Um, and so these sooty manga bees, these are small monkeys that people sometimes use for food. And so this probably got into people when people were um, using wild animals as a source of food. And as they were cleaning the animals, some of the viruses from the animal wound up in people. And so we see the most common types of HIV, HIV-2, as coming from that. And even the other HIVs, the chimpanzee HIVs that are similar to HIV-1, they're less common. But since they come from a variety of sources, we also think it's probably these were used as sources of food. So here's an example what we can kind of tell where things came from and a source of disease. We're doing the same thing right now with COVID and trying to answer questions about where COVID came from. And it turns out COVID is very similar to viruses in bats. And so we can look at the sequence of the COVID genes and we can tell we're pretty sure, okay, this came from an accidental transmission from humans being in contact with bats. Maybe food, maybe another way. Okay, we'll stop there and